So these ants, it's a little bit different. So they're going to be zombie-like. So within a few hours, maybe a day, the ant will begin to behave abnormally.
Something strange is happening in laboratories around the world. Biologists studying a new fungal strain have begun reporting results they can't explain.
This fungus is altering the behavior of the ant to allow the dispersal of their spores that can go in the environment and colonize other ants.
Abnormal nerve activity, sudden frightening behavior changes, and a pattern that looks hauntingly familiar. For decades, this phenomenon was known only in insects like ants, where a fungus could seize control of its host. Now, early data suggest it may no longer be limited to them. The terrifying question is, how many of us has it already begun to control? The parasite that rewrites nature. In the heart of the world's rainforests, something horrifying happens every single day, hidden under the leaves where sunlight barely reaches. Tiny ant colonies march in perfect order, unaware that among them lurks one of nature's most disturbing predators. It is not a snake, not a spider, but a fungus. Its name is Ophiocordyceps unilateralis, and what it does is unlike anything else on Earth. This fungus does not just infect its host, it takes control of it completely. It all begins when a single spore lands on the exoskeleton of a carpenter ant, a species known as Camponotus leonardi. These ants live high up in the trees, building aerial pathways of leaves and branches. But sometimes, one of them climbs down to the forest floor. That small mistake can mean death. The moment the spore touches the ant, it begins its silent invasion. It germinates, spreading microscopic filaments called hyphae that push through the ant's armor-like shell. The fungus releases enzymes that dissolve the hard outer layer, while mechanical pressure forces the filaments deeper. Within a day or two, they reach the ant's inner body fluid, called hemolymph. From there, the infection spreads fast. Once inside, the fungus begins to grow like an invisible web. It does not need to enter the brain. Instead, it wraps itself around the muscles, forming a network that controls every movement. Scientists studying infected ants found that the fungus releases chemicals that act like false signals to the muscles. These chemicals override the ant's natural nerve impulses. It is as if the fungus has stolen the remote control to the ant's body, while the ant's brain is still aware but completely unable to resist. Researchers describe it as the ant being a prisoner inside its own body, watching itself move against its will. The next stage is even more disturbing. About a week after infection, the fungus forces the ant to leave the safety of its colony. It makes the ant wander away, down the tree, toward a location that fits the fungus's exact growth requirements. This destination is not random. Studies have shown that nearly every infected ant ends up about 25 centimeters above the ground, on the underside of a leaf, and usually on the north side of the plant. The air there is warm, humid, and perfectly stable, with moisture levels between 94 and 95%. Those are the exact conditions that help the fungus thrive. When the ant reaches this ideal spot, it performs the final act of its life. It locks its mandibles around the central vein of the leaf and clamps down hard. This is known as the death grip. The fungus then kills the host and begins to consume what remains of it from the inside. A few days later, a dark stalk bursts through the top of the ant's head. At its tip forms a capsule filled with thousands of new spores. When the wind or rain hits the leaf, those spores fall to the forest floor, ready to infect new ants passing below. Researchers at Penn State University and Harvard University used advanced 3D microscopes and machine learning software to map how the fungus grows inside the ant. Their findings, published in 2017 and 2019, confirmed that the fungus creates a highly organized structure that functions almost like a nervous system. It connects muscle to muscle, forming a communication network throughout the ant's body. The precision of this system is what allows the fungus to move the ant like a puppet. For biologists, this discovery was more than just a shocking example of nature's cruelty. It showed that microorganisms can do something once thought impossible. They can manipulate behavior, not through intelligence or thought, but through chemistry and evolution. If a fungus could control an ant so perfectly, what would stop a similar organism from adapting to other animals? What if it could survive in mammals, or even in humans? The closer researchers looked at the strange relationship between fungus and host, the more unsettling questions began to surface. Could something like this already be happening in us, hidden in ways we do not yet understand? That question drove biologists to begin searching human samples, and what they found next left them stunned. When AI looked closer, new patterns in human samples. In early 2025, a quiet collaboration began among laboratories in Singapore, Brazil, Germany, and the United States. The group consisted of microbiologists, neurologists, and data scientists who specialized in infectious diseases that blur the line between the body and the mind. Their goal was simple at first: to use new artificial intelligence tools to map hidden microorganisms in human tissue. The project started as part of a broader global health study that examined the relationship between environmental fungi and unexplained neurological disorders. The team began collecting cadaveric nerve and brain samples from people who had died from unknown neurodegenerative conditions. Every specimen was processed under sterile conditions and scanned using deep learning microscopy, a technique that allows AI systems to detect patterns invisible to the human eye. Alongside this, they used metagenomic sequencing, which decodes all the genetic material inside a sample to find DNA from bacteria, viruses, or fungi that standard tests might miss. These technologies are real and commonly used to detect new or rare pathogens. As the data poured in, one pattern kept appearing. Several nerve tissue samples showed thin, branching filaments that did not match any known human structure. At first, the team thought these were contaminants, perhaps fungal spores from the environment or tissue-handling artifacts. But when the same formation appeared across samples from 3 different continents, the researchers knew this was not coincidence. The AI model classified the structures as non-human biological filaments, and further analysis showed that their architecture closely resembled the filaments of Ophiocordyceps unilateralis, the same fungus that controls ants. Alarmed, the team ran genomic comparisons between the unknown filaments and a database of fungal genomes. The computer returned a result that froze everyone in the room. The unidentified organism's DNA shared about 90% similarity with the gene group responsible for motor protein control in Ophiocordyceps. These are the genes that allow the fungus to attach to muscles and send chemical signals that override movement. It was not a perfect match, but it was far closer than anyone had ever seen in a human-related sample. The next step was chemical analysis. Using mass spectrometry, the scientists isolated several compounds secreted by the mysterious cells. These compounds behaved like tryptamines, a family of chemicals that include serotonin and other neurotransmitters that affect mood and motion. The lab found that these fungal chemicals could bind to serotonin receptors in cultured human nerve cells. When that happened, the electrical signals inside those cells changed dramatically. The nerves fired more frequently and stayed active longer than normal, which could theoretically alter motor coordination or emotional response. As more samples were processed, a strange pattern began to emerge. The AI consistently highlighted dense concentrations of these fungal structures along the vagus nerve. The vagus nerve is one of the body's longest and most important nerves, connecting the gut, heart, and brain. It plays a key role in what scientists call the gut-brain axis, the system that allows the digestive system and brain to communicate. This was an alarming location for any foreign organism to occupy. It meant the fungus was not simply invading random tissue. It was forming networks along the same route that the body uses to regulate emotion, hunger, and stress. The researchers worked quietly. They did not release public statements. They wanted to repeat the results before raising global concern. By mid-2025, they had verified that 22 cadaver samples, taken from 6 different regions, showed identical filament structures and chemical activity. No existing database could identify the organism, and no previous pathogen had ever been seen occupying the vagus nerve in this way. To many of the scientists involved, the finding was deeply unsettling. It hinted that something fungal might be living inside human nervous tissue, not as a typical infection, but as a network capable of chemical communication. The question was whether this network could influence behavior, even slightly. To answer that, the team turned to the only known microbe already proven to change how mammals think and act. It was not a fungus at all, but a parasite, and it had been quietly living inside a third of the human population for thousands of years. The parasite already inside us. The culprit is a microscopic parasite called Toxoplasma gondii. It is one of the most successful parasites on Earth. Most of those infected never know it, but the changes it causes inside the brain have shocked scientists for decades. The life of Toxoplasma gondii revolves around one animal, the cat. Cats are its primary hosts, meaning the parasite can only reproduce inside a cat's body. To reach a cat, Toxoplasma must first travel through other creatures known as intermediate hosts. These can include birds, mice, and sometimes humans. The parasite's strategy is simple but incredibly effective. It hides inside the tissues of these hosts, waiting for one of them to be eaten by a cat, where it can finally complete its life cycle. Infection begins when a person or animal ingests tiny, tough-walled eggs, known as oocysts. These can come from undercooked meat, contaminated soil, or contact with cat feces. Once inside the body, the parasite moves through the bloodstream and forms microscopic cysts in muscles, eyes, and especially in the brain. In healthy people, the immune system suppresses the infection, keeping the parasite in a dormant state that can last for years or even a lifetime. But even when inactive, Toxoplasma gondii does not stay silent. Scientists first noticed its strange influence in rodents. Normally, a mouse's survival depends on its fear of cat scent. A whiff of cat urine should send it running for safety, but infected mice behave differently. They lose their natural fear and in some cases are even drawn toward the smell. This suicidal attraction dramatically increases the chance that a cat will catch and eat them, which is exactly what the parasite needs. By altering its host's brain chemistry, Toxoplasma manipulates behavior to ensure its own survival. When researchers discovered this, they began to wonder if the same thing might happen in humans. Over the past 2 decades, studies in the Czech Republic and Denmark have tracked thousands of people to find out. The results were unsettling. Infected individuals consistently showed slower reaction times and were 40 to 50% more likely to be involved in traffic accidents. Other studies linked infection to personality changes such as higher risk-taking and impulsive behavior. Infected men were found to drive faster, take more physical risks at work, and report stronger attraction to danger, while infected women showed higher sociability and lower fear of confrontation. Some participants also scored higher on impulsivity tests, reporting sudden mood changes and poor judgment under stress. The pattern was too strong to ignore. Deeper research into brain imaging and chemistry revealed how the parasite does this. The cysts tend to cluster in the amygdala, the region of the brain that processes fear and emotion. Within that area, Toxoplasma affects the production of dopamine, a neurotransmitter that regulates motivation and reward. Too much dopamine can disrupt normal emotional control, leading to reckless or unpredictable decisions. Several meta-analyses have also shown an association between Toxoplasma infection and schizophrenia, suggesting that the parasite's chemical interference may push vulnerable individuals toward mental illness. Despite these findings, Toxoplasma gondii rarely causes visible damage. It does not destroy brain tissue or cause inflammation like many infections do. Instead, it works quietly, changing the chemical balance that controls how the brain interprets fear, pleasure, and reward. This ability to manipulate behavior without causing physical harm is what makes it so remarkable. It proves that a microorganism does not need to attack the brain directly to change the mind. It only needs to alter the signals that control how we respond to the world around us. For years, scientists assumed that only simple animals like rodents could be influenced this way. But human studies have shown that the boundary between psychological and biological control is thinner than anyone once believed. If a single-celled parasite can alter emotions and behavior through chemistry, what about more advanced organisms? The question soon became impossible to ignore. If a tiny protozoan can rewire fear and attraction through chemical signals, could a complex, heat-resistant fungus evolve to do something even more powerful inside us? That possibility is what led biologists to begin examining a series of strange new infections that appeared to defy all known patterns. The first human case files. In late 2024, doctors in northern Thailand began to notice a pattern that made little sense. Several patients from rural villages arrived at regional hospitals complaining of sudden muscle spasms, dizziness, and periods of strange, uncontrollable movement. Some described waking up in the middle of the night with an overwhelming urge to walk or climb, even when they knew it was dangerous. A few were found outside their homes at dawn, gripping trees or fences as if their bodies refused to let go. Most of these patients were farmers or field workers who spent long hours in humid environments, where exposure to soil and decaying vegetation was part of daily life. At first, the cases were treated as neurological disorders. Doctors suspected early signs of epilepsy or chemical poisoning from pesticide exposure. Routine tests showed nothing unusual. The patients had no signs of brain infection, and their reflexes remained normal when they were conscious. Yet, during their episodes, their bodies seemed to move on their own. The spasms were coordinated rather than random, resembling the controlled but unnatural motion seen in certain movement disorders. Local physicians were puzzled. 2 months later, a similar cluster of cases appeared in the Brazilian state of Amazonas. The symptoms were nearly identical: muscle contractions, loss of voluntary control, and compulsive behavior such as climbing and repetitive gripping. In both Thailand and Brazil, the hospitals treating these patients noted that antifungal medications, which normally stop infections like Candida, had little to no effect. Blood samples showed mild inflammation, but no bacteria, viruses, or known fungal spores were identified through standard lab tests. With no answers, the samples were sent to an international research network in Geneva. There, scientists used advanced tissue imaging and deep genetic sequencing to analyze nerve biopsies. What they found was unlike anything they had seen before. The nerve tissue contained long, branching fungal filaments, known as hyphae, that wrapped tightly around the patient's motor neurons. Yet the gray matter of the brain remained untouched. This pattern was eerily similar to the Ophiocordyceps unilateralis fungus that infects ants. It was as if the organism had learned to copy its insect-targeting strategy inside human tissue. Further examination revealed that the fungal cells were alive and metabolically active. They appeared to secrete a series of complex molecules that interfered with normal nerve signaling. Electrical tests showed that motor neurons surrounded by these filaments fired in irregular bursts, creating involuntary contractions in muscle fibers. The patient's nervous systems were not completely hijacked, but they were no longer fully under their own control. The discovery caused quiet alarm within the scientific community. The team classified the organism under a temporary name, Ophiocordyceps hominis, meaning human Ophiocordyceps. Only 12 cases were confirmed during that period, but the possibility of more undetected infections could not be ruled out. Field investigators traced most of the patient's work environments to damp agricultural areas rich in decaying plant matter, the same type of environment where traditional Ophiocordyceps species thrive. Soil and air samples from those regions later tested positive for spore-like particles genetically related to the fungal family, suggesting a potential soil spore origin. Although no deaths occurred, the nature of the infection made containment urgent. All confirmed patients were isolated in controlled medical units. Disinfection procedures were intensified, and travel restrictions were quietly imposed in the affected regions. Scientists feared that if the fungus could survive at human body temperature, it could spread quickly through air or contact with contaminated soil. To understand how plausible this was, experts looked to the history of other fungal species that had crossed into humans. The most cited example was Candida auris, first discovered in 2009. It had evolved the ability to withstand higher temperatures, making it one of the first fungi to thrive inside the human body after adapting to global warming. The same type of evolutionary leap could explain Ophiocordyceps hominis. By the start of 2025, the team released an internal report labeling the organism as a potential emerging pathogen under restricted surveillance. The public was not informed, and most media outlets never learned of the investigation. The scientists involved feared panic and wanted more data before making any announcement. But one fact was clear: this was not a coincidence. The fungus was following a known evolutionary path, adapting to heat, changing its biology, and finding new hosts. That same adaptation, the ability to survive in the warmth of human tissue, became the key focus of every subsequent study. Researchers soon realized that climate itself might be driving this transformation, giving rise to fungi that no longer die in heat but thrive in it. The mutation that loves heat. When scientists began analyzing the genetic makeup of Ophiocordyceps hominis, one detail stood out immediately. The fungus appeared to have developed a powerful resistance to heat. For decades, temperature had been one of humanity's best defenses against fungal infections. Most fungi thrive in cooler environments and die when exposed to the internal warmth of the human body, which averages 37 degrees Celsius. But as the planet has warmed, that protection has started to fail. Across the last 2 decades, researchers have documented a steady rise in fungal species capable of surviving at higher temperatures. The most alarming example is Candida auris, first identified in 2009. Within only a few years, it spread to more than 40 countries and became one of the most difficult infections to treat. Hospitals around the world reported outbreaks of Candida auris that resisted multiple antifungal drugs. Studies later confirmed that the fungus's sudden appearance was likely linked to climate change. By gradually adapting to warmer outdoor temperatures, it accidentally gained the ability to survive inside the human body. In laboratory settings, experiments have shown how this transformation happens. When fungal colonies are exposed to slowly increasing temperatures, they evolve a defense system based on special molecules called heat shock proteins. These proteins protect the organism's cells from damage caused by heat stress. In several species, scientists have observed this adaptation occurring in fewer than 100 generations, which can take less than a year. This speed of evolution shocked many researchers and proved that fungi are not as static as once believed. The genome of Ophiocordyceps hominis seemed to reflect the same adaptation. Its cell walls contained elevated levels of heat shock protein genes, suggesting that it could comfortably live at 37 degrees Celsius and possibly endure short periods of fever without dying. This finding meant that the fungus could survive the natural defenses of the human immune system, which usually relies on raising body temperature to kill pathogens. The possibility of transmission worried scientists even more. Environmental studies indicated that the spores could remain viable in the air for up to 48 hours and on surfaces for more than 72. That is similar to the resilience of Aspergillus, a fungus already known for causing severe respiratory infections. The spore size of O. hominis was small enough to be carried through air currents, dust, or moisture droplets, especially in closed or humid indoor environments. Contaminated produce or soil particles might also carry the fungus from one region to another. If these conditions held true, it meant that the fungus was no longer limited to a few tropical zones. It could survive transport, resist heat, and potentially establish itself in any warm, damp location on Earth. Once it was clear that the fungus could live inside the human body, researchers turned their attention to something even more disturbing. They wanted to know whether it could do what its ancestors once did to ants, interfere with behavior, and early laboratory results revealed something deeply unsettling. The behavioral switch. By early 2025, researchers following the Ophiocordyceps hominis outbreak began noticing that the infection was doing something no fungus had ever been observed to do. It was not just affecting muscles or nerves anymore. Patients who had overcome the initial spasms started showing changes in judgment, fear, and self-control. Some reported moments of euphoria or bursts of aggression without warning. Others described an unshakable feeling of confidence, as though ordinary caution had been switched off. These shifts were small at first, but consistent enough to suggest something far more serious. The fungus was not just living inside the body, it was beginning to interfere with the mind. Fungal biologists already knew that some species could produce chemicals capable of influencing human consciousness. Psilocybe cubensis, the mushroom used in hallucinogenic drugs, produces psilocybin, which binds to serotonin receptors and alters mood and perception. Another well-known species, Claviceps purpurea, can release ergot alkaloids that trigger convulsions, delirium, and hallucinations. These effects are not psychological, but biochemical, meaning they happen through direct interference with brain chemistry. The research team studying O. hominis began to suspect that it had developed a similar capacity. When scientists analyzed fungal cultures taken from infected nerve tissue, they detected compounds that acted like serotonin analogs, chemicals capable of binding to the same receptors that regulate impulse and emotion. These molecules were faint but persistent, and when added to samples of healthy human neurons in the lab, they caused the nerve cells to fire at irregular intervals. Electrical recordings showed bursts of activity similar to those seen in people experiencing panic, intoxication, or extreme stress. The cells did not die, they simply became erratic, firing even when no signal should have been present. Soon after, MRI scans from infected volunteers confirmed what the chemistry implied. The scans revealed a 30% decrease in connectivity between the prefrontal cortex, which controls decision-making, and the amygdala, which governs fear. This meant emotional signals were no longer properly balanced by reasoning. The infected patients described feeling unusually fearless, restless, or excited, even in dangerous situations. Their behavior looked normal from the outside, but their sense of risk and reward had been rewired. Doctors and public health investigators began linking these neurological findings to events unfolding in several affected regions. In small towns where fungal spores had been detected, reports of reckless or impulsive behavior started to rise. Hospitals saw more injuries from stunts, climbing attempts, and speed-related accidents. Police departments logged a sharp increase in fights and sudden acts of aggression. The scientists named this stage of the infection the behavioral switch. It did not make people lose consciousness or hallucinate. Instead, it subtly altered how they processed fear and excitement. Those infected did not feel controlled or possessed. They simply acted without hesitation, driven by impulses that felt natural. This made containment far more difficult. Infected individuals were unlikely to report their symptoms because, to them, nothing felt wrong. The discovery forced ethicists and legal experts to confront an uncomfortable question. If a person under the influence of this organism committed a crime or caused harm, could they truly be held responsible? How would anyone determine where human decision ended and fungal chemistry began? Those questions would soon become impossible to ignore because what happened next would prove that the fungus is no longer an isolated threat. It is becoming a global event. It's spreading, and there's no containment. Recently, biologists and disease analysts monitoring Ophiocordyceps hominis began issuing quiet but serious warnings. What had once been described as a limited tropical anomaly is now showing up in soil, dust, and air samples from different parts of the world. The findings are not proof of an outbreak, but they are enough to worry everyone involved. When identical genetic fragments began turning up in urban air filters and produced shipments, the pattern became impossible to dismiss. The fungus had found a way to travel farther than anyone had expected. So far, no nation has declared a public emergency, but epidemiologists are already running projection models to predict what could happen if the trend continues. Simulations show that if global humidity levels rise even slightly, the spores could stay active for up to 2 full days in the air and nearly 3 days on surfaces. That means they could hitchhike across borders in cargo containers or ventilation systems. A single contaminated shipping batch could carry viable spores halfway across the planet before anyone noticed. The immediate danger is a gradual, unnoticed influence. The fungus seems to work by shifting the balance of behavior, tiny changes in decision-making that add up over time. Public health experts warn that a mild infection might first appear as an increase in impulsive or reckless behavior, especially in densely populated cities where the spores could spread silently. One scenario suggests that as more people lose their natural fear responses, accident rates could rise in transport and construction sectors. More drivers might take unsafe turns or speed through traffic. Workers in high-risk industries might ignore safety procedures. One misjudgment could cause a chain reaction that could turn into a nationwide safety crisis. The consequences would not stop at work or on the road. Sociologists predict that even a small population-level increase in impulsivity could lead to spikes in aggression, gambling, and violent outbursts. Researchers already note that reduced connectivity between the brain's fear and control centers could make people more reactive, more confident, and less empathetic. That combination could destabilize entire communities. Street altercations, domestic violence, and riots could become harder to contain if collective inhibition continued to weaken. Economists and policy analysts are equally concerned about how such behavioral shifts could impact governance and global stability. In markets driven by emotion and risk-taking, a drop in caution could inflate speculative bubbles or crash industries overnight. Political decisions might become less measured, driven by emotion rather than reason. The potential agricultural impact adds another layer of danger. If spores contaminate crops, they could quietly disrupt the food supply chain. Experts warn that the fungus might spread through soil and humidity-controlled greenhouses, particularly in tropical regions. Even trace contamination could prompt mass recalls and food shortages. Regions dependent on export agriculture would face economic strain, while wealthier nations would compete for unaffected food sources. Efforts to prevent these outcomes are reportedly underway, though still at an early stage. Research teams are quietly experimenting with RNA-based antifungal vaccines that could target the fungus's genes responsible for surviving heat and producing serotonin-like compounds. Others are building AI-driven detection networks capable of identifying fungal genetic markers in public ventilation systems. But these projects are years from deployment, and scientists admit that containment would be difficult once environmental contamination became widespread. What makes this even more disturbing is that almost none of this information is public. Most of the data still circulates only inside academic networks, unpublished research papers, and closed scientific briefings. Leaked reports and anonymous testimony from lab researchers have provided the only glimpses of what is happening behind the scenes. Governments have not confirmed or denied the existence of these findings, citing the need to prevent panic and verify the evidence first. As scientists quietly warn policymakers of these possibilities, one fact remains clear. The fungus does not need to destroy us to win. It only needs to keep evolving, quietly, steadily, and everywhere we breathe. What do you think about this discovery? Comment down below.